Thank you. We're here on EVClub today in the
Yeah.
in the conference center in Lyon, France, with the ISEV2022 meeting. And so you've just presented a poster. I think a very exciting poster that you had sent me some details on before. So I just wanted to ask you, first of all, tell us who you are and where you're working.
Sure. So my name is Michael Witwer. I'm a clinician. I'm an obstetrician gynecologist. I'm doing a PhD based at the University of Edinburgh in the UK. And I'm working on EVs in the prepubertal male reproductive tract and their role in infertility in later life for children with cancer.
Excellent, excellent. So how many ISEV meetings have you been to?
So if we're talking about in-person ones, this is my first, and speaking last as a PhD student, but I was fortunate enough to come and visit online.
Excellent, excellent. All right, well, welcome to ISEV 2022.
Thank you.
So you presented a poster today. Do you want to just give us a very brief overview of your findings?
So we are interested in male infertility, in particular for children with cancer. So we look at young boys who are prepubertal and what we can do to preserve their fertility. So we're able to look at the spermatogonal stem cells and Sertoli cells using a mouse model, and we treat those cells with cisplatin, which is a commonly used chemotherapeutic to generate EVs. Mm-hmm. And we've identified very briefly that if you treat Sertoli cells, which creates a protective environment for developing sperm, if you treat those with cisplatin, they release EVs, which induce cell death in treatment-naive Sertoli cells. And we've not observed this before. And we think that's important because if there's an additional impact from EVs in the testes from chemotherapy, it means you might be able to modulate that effect to try and reduce infertility in later life. It's important to remember, if a child has cancer, you can't withhold the chemotherapy. So it's really important that we find other mechanisms through which this damage is occurring.
So is this because the cisplatin is being transferred, or is this an indirect effect?
So that's a really good question. So we actually looked to see if there was cisplatin in the EVs because we thought this would be really interesting if we found another mode of cisplatin delivery. But from our mass spec data, we don't think the EVs are carrying cisplatin. So we think it's something else in there.
And do you know what it is?
So we don't know, but I've been very lucky. I've just been able to visit Dr. Shannon Stott's lab at MGH in Harvard to do some sequencing. So we've done some RNA sequencing and going to do some mass spec back in Edinburgh to really figure out what's inside the EVs which are causing these effects.
Excellent, excellent. All right. So you were just in the poster session.
Yes.
And so did you get any questions that were particularly interesting or that stumped you or that you made you think about something new?
Yeah, so we had a few questions. I think one question that everyone asks is it the EV, or is it just the co-precipitate, which we've not identified? Mm-hmm. We do feel it is the EVs because we can see them going inside the cells. We were fortunate to have an optic-nan imager so we can track the EVs moving in the cell. And we've also done that on another machine as well.
To tell the, I mean, the audience doesn't know what the Oxford Nano imager is. What is this?
So the Oxford Nano imager, or the ONI, kind of sponsored everyone's line.
Oh, there we go.
It's a really cool machine that we're able to use in Edinburgh. It lets you do a few things. So you can effectively zoom in a cell. We're able to see individual vesicles moving within inside the cell, and we can track them moving in real time and the speed and directionality of those EVs. We can also do single EV D-storm imaging for CD9, 63, and 81s like that. The whole trilogy, isn't it, really, of the EV markers. And so we were able to use that equipment as well as some live cell imaging on a machine called IncuSite. So it just takes pictures of your 96 well plates. And we're able to do that to look at EV tracking and cell death following incubation with our EVs.
Excellent. All right. Yeah. So where do you want to take this project next? You told us a little bit about the RNA sequencing and.
Yeah. So what I would like to do is I'd like to see what's inside the EV. And if we can find out something interesting, we want to put those EVs back on the cells and then try and interrogate the cells. Because if we can say, well, this unique marker is in the cisplatin-derived EVs, it then makes this change occur in the cell. We're not sure what that would be yet. And we've seen the phenotypic impact on the actual cell death. We feel there's a nice story there as well. What I'd also like to do if we did more sequencing data is to compare that to the relevant human tissue. So people have looked at the impact of chemotherapy on various different tissues, and if we were able to find a signature that was changed in our model and the relevant human tissue, I think everyone in human tissue assumed it's through chemotherapy, but we might be able to say, well, actually, we see a similar change when we incubate EVs only, which would be really exciting.
Very good.
To pick out those mechanisms.
Perfect. All right. Well, Mike, thanks so much for joining us here and telling us about your work. And we wish you all the best with it, and good luck.
Thank you so much.